Welcome to today's Explainer. We are about to take a seriously fascinating journey, starting all the way back in the bubbling hot springs of early Earth and zooming all the way down to the hidden microscopic boundaries of your very own cells. We're going to break down the fundamental building blocks of life today, and I am incredibly excited to uncover exactly how life protects and structures itself. Let's dive in. All right, to kick things off, I want you to imagine drawing two tiny dots on a piece of paper. As you bring those dots closer and closer together, there's a specific physical point where they just kind of blur into a single dot to our naked eye. That distance, it's exactly zero point 0.1 millimeters. That physical wall is what scientists call the limit of resolution. It's basically the absolute maximum ability of the human eye to see two very close objects as completely separate and distinct. Because our naked eyes hit a physical wall at this zero point 0.1 millimeter limit, humanity had to figure out a way to zoom in if we ever wanted to actually understand the fundamental units of life. Okay, Section 1, Origins of Life. We're tracing life all the way back 3.5 billion years. Think about some of the most extreme environments on our planet, similar to the nearly boiling hot springs of the Puga Valley. In harsh environments just like that, scientists actually discovered rapid calcium carbonate deposits. Now, this is incredibly significant because these deposits likely shielded early organic molecules from those harsh conditions and harmful radiation. Ultimately, this helped form the very first protective cellular membrane. We're talking about the barrier that literally separates life from non-life. Moving right along to Section 2, Beyond the Naked Eye. Let's look at how we overcame that zero point 0.1 millimeter limit. Just look at this incredible technological leap. On the left, you've got a classic light microscope, which functions a lot like the one Robert Hooke used way back in 1665 to discover cells, giving us about 200 to 300 times magnification. But on the right, we have a modern electron microscope. Instead of light, it uses a beam of electrons, which is a total game changer because it lets us see fine cellular structures down at the nanometer scale. So once we're zoomed in, how do we actually know the size of what we're looking at? Well, it's a pretty simple, elegant process, actually. First, you just measure the visible field diameter. Let's say it's 5 millimeters, which is 5,000 micrometers. Then, you literally just count the number of cells lined up across that diameter. By simply dividing that field diameter by the number of cells, boom, you can easily estimate the actual size of a single cell. Which brings us to Section 3, the universal boundary. Let's talk about the fluid mosaic model of the cell membrane. This is what scientists call the fluid mosaic model, and it details exactly how this outer defense functions. Think of it as a flexible, moving layer of fats. They have these water-attracting heads and water-repelling tails, and because they can move and flip around, it's considered fluid. And scattered throughout all of this are protein gatekeepers. They're arranged just like little mosaic tiles, and their whole job is to selectively decide what gets to enter and exit the cell. All right, Section 4, the potato experiment. Let's test the selective membrane. We really have to ask ourselves, how exactly do these microscopic gatekeeper proteins react when the environment outside the cells suddenly changes? Like, what happens if that outside environment suddenly becomes heavily concentrated with salt or sugar? Well, here is the visual proof of those membranes hard at work. We took a potato and cut it into two equal pieces. When one piece is placed in beaker A with just plain water, and the other goes into beaker B with a 20% salt solution, those cellular membranes immediately spring into action to try and balance out their internal environments. Let's see what happens. The result we get after leaving them for just an hour is pretty striking. In plain water, that potato piece actually swells up and gains weight. But in the heavily salted water over in beaker B, the potato piece noticeably shrinks and loses weight. So what we're seeing here reveals the rules of osmosis in action. By the way, this is exactly the reason drinking ocean water dehydrates you. If the concentration is equal, which we call an isotonic solution, nothing changes. But plain water, that's hypotonic, meaning it has a lower concentration of solutes than the inside of the cell. So water just rushes in, making the cells swell up. On the flip side, the hypertonic salty water has a higher concentration, so it actually pulls moisture out, forcing the cell to shrink. Section 5, Plant versus Animal Cells. This is a crucial structural difference. Notice the stark contrast between these two. The plant-based onion cells on the left, they're perfectly box-shaped, super regular, and neatly arranged. But then look at the animal-based human cheek cells on the right. They're totally irregular, softer, and completely lack a uniform structure. The reason for this massive visual difference really comes down to this. Plants, along with fungi and bacteria, possess an additional, rigid protective layer outside their plasma membrane, called the cell wall. Animal cells entirely lack this cell wall. They only have that thin, fluid membrane we talked about earlier. And because animal cells, like our cheek cells, only have that flexible plasma membrane and no rigid wall to hold their shape, they are incredibly susceptible to losing water. If you drop them into a concentrated sugar or salt solution, those poor animal cells will rapidly lose water and shrink right up, without a cell wall there to provide structural support. So, I want to leave you with a final real-world puzzle based on everything we've just covered today. If you take soaked mung beans, which, remember, are plant cells with rigid cell walls, and you put them in a highly concentrated solution, what exactly will happen to their cells? Knowing what you now know about osmosis and cell walls, think about how they're going to react. Keep asking those questions, and I really hope this explainer has sparked your curiosity about our incredible microscopic world.